Holly Bridge from Oxford University will talk on the role of neurochemistry.
Am I good? Am I good to go? Can you see my slides?
Yes, we're good.
Okay, excellent. Yeah, well, thank you very much for the invitation to speak, and I'm sorry that I can't be there in sunny California because I'm sure it's a lot sunnier than it is here. But yeah, so I'm going to talk about neurochemistry, and we might come back to that idea about what the difference between what BOLD tells you versus what neurochemistry can tell you. So magnetic resonance spectroscopy is essentially a technique that we can use using the MRI scanner, which allows us to get a measure of chemicals within the brain. So in this diagram, essentially, we've got a square of brain tissue, and what we're doing is we're essentially summing everything that's within that area of the brain. And what we do is identify metabolites using knowledge of their chemical properties. So the chemical properties will determine the position along this chemical shift axis. So what you can see is that these different metabolites that have different arrows, so different parts of those metabolites are at different positions on this axis, and basically by clever integration, we can work out what the concentrations of these different metabolites are within this sort of chunk of brain. And for what I'm interested in, particularly, it's GABA and glutamate as the sort of main inhibitory and excitatory transmitters that we're interested in measuring. So why is it important to measure neurochemistry? Well, I've got a few points here. So, when we think about what's going on in the brains, we often think about, action potentials, propagation. But of course, as we all know, it's actually the chemical transmission at synapses is which gives us the computational power for our brains. The plasticity that allows us to learn and to adapt to our surroundings relies on both these excitatory, glutamatergic, in this case, and inhibitory synapses. And we know that the disruption of the balance between excitation and inhibition is implicated in various different clinical conditions, things like epilepsy. And then sort of, I guess, relating to what Ione was just asking about or made a point about is that, the BOLD signal does not tell us everything, and that there is sort of growing evidence that actually the changes in neurochemistry may actually reflect things like learning better than we would see in a BOLD signal. So what I'm going to talk about, what I really wanted to give you an idea about is the sort of how we use the MRS. So two different research areas that I've been involved in. So the first thing I'm going to talk about is GABA in ocular balance, so hopefully sort of relating to the previous session. And then in the second part, I'm going to talk about neurochemistry when we've got damage to the primary visual cortex. So this is from a paper by Mensch, but the idea that inhibition is really important for intraocular balance. So if we imagine the signals coming from the two eyes, basically what we want to happen at the output of this is that we make a binocular neuron that gets equal input from the two eyes. And one of the ways in which we can do that is this use of inhibitory, sort of mutual inhibition, where the right eye, information from the right eye neuron is going to activate this inhibitory interneuron, which then inhibits the left eye's neuron. And essentially, if the firing in these two neurons is equal, then they should have equal amounts of inhibition. And what we end up with is this nice balanced binocular neuron. Obviously, in the previous session, we heard a lot about when ocular balance goes wrong. And the idea is that what might happen, one way to explain how that becomes a sort of brain problem is that if we have reduced input from one eye, essentially we get less firing in this neuron here, which means that this inhibitory interneuron gets excited less. So we've got less inhibition of this neuron, which means that actually by the time we get to the binocular neuron, it's activated predominantly by this left eye neuron. So this kind of implicates inhibition, and suggests that when we get this sort of disruption of balance, it may be because our inhibitory system is not working correctly. So one way in which we could, so I'm not actually going to talk about sort of amblyopia and actual disruption to eye dominance or clinical types of disruption, but I'm just going to talk a little bit about eye dominance. So this is the way in which we measure eye dominance is essentially by using binocular rivalry. So all we're going to do is we are going to present dichoptically different stimuli to the left and the right eye, and we're going to measure what we call the mean phase duration. So the ratio of the time in which we see the stimulus presented to the left eye divided by the time that we see it presented to the right eye. And those sort of mixed periods where you kind of see the two stimuli sort of merging together, we will just ignore that. So basically the people are going to report when they're seeing the left eye versus the right eye stimulus. So my interest in this actually started when Claudia Lunghi, pictured here, came to my lab. So with Concetta and the rest of the lab, they've been looking at this idea about what happens when you have monocular deprivation, in particular looking at the plasticity that you can get in adults. And these are adults with normal vision. So they used this rival stimulus, and they were interested in this paradigm where you have 150 minutes of monocular deprivation, and you test people's binocular rivalry before and after that deprivation. And what they found was that if you look at the change in the amount of time that they perceived the stimulus to the deprived eye after deprivation, it actually increased significantly, whereas the amount of time perceived by the non-deprived eye actually decreased. And if you looked across time, so 3 minutes after you remove the eye patch, your perception is almost completely dominated by the eye that has been deprived, and then the amount that your sort of balance then returns over about 90 minutes. But you can see for the first 15 minutes, much of your perception will come through that eye that had been deprived. So this was an interesting finding. So the reason that Claudia came to my lab was to see whether we can actually see whether there is a neurochemical basis for this change. So we were really fortunate that, well, so firstly, because it was back in sort of 2013, we had a 7 Tesla scanner, which gives us better resolution of the neurochemicals. And when Claudia came and presented this study to our lab, and it happened that this guy here, so Uze Amir, he'd just arrived in Oxford and he specializes in spectroscopy. So fortunately, we were able to sort of use his expertise and set up a study that basically allowed us to use their paradigm. So we test the binocular rivalry for 5 minutes at the beginning of the study. Then we get people to go into the scanner for about an hour while we collect the spectroscopy data. Then they do the 2.5 hours of monocular deprivation. And during that period, they actually walked around our center. They actually kind of entertained themselves. And then we brought them back into the lab. We tested their binocular rivalry again before sticking them back in the scanner to repeat the session. So this is just showing the sort of behavioral data, and what you can see is, so this is the binocular rivalry results, fairly balanced between the two eyes. So the blue dots are people who have the monocular deprivation, and then we had some control subjects who did the scanning and the binocular rivalry but didn't actually have the monocular deprivation, so they're in red. So you can see baseline, everyone is fairly balanced. After deprivation, you can see that all the blue dots are kind of shifted underneath this line, suggesting that they now spend more time perceiving through the deprived eye than the non-deprived eye. And again, that's sort of summarized here. So instead of a sort of balance of 1, so the ratio equaling 1, you can see it's now spread between sort of 1 and about 4. So we get the behavioral effect. So what happens then with the GABA? So this is where we measured from. So we put that voxel, so 2 by 2 by 2 centimeters. So it's quite a big area. But V1's nice to measure from because obviously you've got grey matter from the 2 hemispheres. So you get a lot of sort of brain tissue in that region. And then as a control voxel, just to check we don't get a general effect throughout the brain, we also took the posterior cingulate, which is also on the midline with a similar amount of grey matter within the voxel. And essentially the participants lay down in our 7T scanner, and we did a variety of different scan conditions, but the one that I'm presenting is just really with the eyes closed, and from V1 and from that control posterior cingulate voxel. So again, this is the voxel that we're taking from, and this is an example spectrum from probably our best subject. And what you can see is when we model the GABA from that spectrum, you can see that at baseline, the GABA, the amplitude of the GABA signal is considerably higher than we find after the deprivation. So in that subject, you can actually see this reduction of the GABA signal. So what happens over the population? Well, you can see when we look at the concentration of GABA at baseline, it's significantly higher than at deprivation. However, we know that the neurochemicals can sort of vary over the course of the day. So, of course, because there's about 3 hours between these two scan sessions, there's a possibility that that's just something that happens because of the time of day. So that's why we had this control group, and we had a control voxel as well. So what you can see is this is now the ratio of the change in GABA concentration. So this is the deprived concentration of GABA divided by, concentration of GABA after deprivation over before deprivation. So you can see that's below 1 with the ones that are deprived in V1. In the posterior cingulate, you can see it's not really significantly different from 1, not surprising. What's actually interesting is that if you look at the GABA concentration in V1 at that, it was basically, I think it was 2 p.m. and 5 p.m. And what you can see is actually, if you don't have the monocular deprivation, you get an increase in the GABA, probably due to that time of day effect. So in fact, that time of day effect was working against us to find that reduction in GABA. So we're fairly confident that the monocular deprivation does reduce the GABA in the sort of primary visual cortex. But what we can also do is actually look at how the change in GABA varies with the behavioral effect. And what you can see is essentially the greater the difference, or the greater the change in your eye balance, the greater the change in your GABA. So these two are highly correlated, where the lower your GABA, or the bigger the reduction in your GABA, the more your deprived eye dominates the signal. So it looks like we've got this, not only do we get the drop in GABA, but it also correlates with the behavioral results. Okay, so that's really shown that we can kind of interfere with the GABA signal if we have this ocular deprivation. But what about in the sort of natural eye dominance? So what does GABA play a role in our eye dominance? So not in terms of amblyopia, but just within the population where we do get this variability. So this is work done predominantly by Bettina Ip in my lab. And again, it's used in the binocular rivalry paradigm to measure eye dominance. So what you can see here is this is just the amount of time that participants perceive through their dominant versus non-dominant eye. And you can see actually these people are quite well balanced. So they're clustered sort of less than, in terms of the difference in these in the eye dominance, it's actually relatively small apart from these ones up here. What we did in this case actually is we used a combined fMRI-MRS sequence, which is nice because it allows us to get both the bold signal and the spectroscopy signal as we flick between. So we're basically going between a blank screen and a flickering checkerboard. So we measure the bold signal as a difference between the checkerboard and a blank screen. But actually, when we're looking for the neurochemical signal, we actually sum across the activity and the blank screen. So we're looking across the whole stimulus. So we do this again. So we look at when the dominant eye is stimulated and when the non-dominant eye is stimulated. And for each of those, what we will do is we'll get this the chemical spectrum, and we'll extract that GABA signal. So what you can see here is if we so we take that so what we're measuring is a sort of intraocular GABA. So that's basically quantified as the GABA concentration when we stimulate the dominant eye minus the GABA concentration when we stimulate the non-dominant eye divided by the concentration of the non-dominant eye. So this is then the relationship between the eye dominance and the intraocular GABA. So obviously, if we've got complete balance, this is going to be 0. So anyone who's got complete balance of their GABA signal will be around here. So you can see that actually we have a big clustering of those participants who have very little in the way of a dominant eye, and they also have very similar levels of GABA when we stimulate the two eyes. And you can see that we do get this correlation, though, as we move away, with the eye dominance moves away from being balanced. I should say, so this was all collected, so this was basically stopped by COVID, which is why we have fewer subjects than we would have wanted. So that's the data that relates to GABA. The question is, so what causes that? So what drives that difference in the GABA response? So it turns out that the change in GABA is actually driven by the non-dominant eye. So there's no correlation between GABA concentration when you stimulate the dominant eye and eye dominance. But when you stimulate the non-dominant eye, what you find is you get this inverse correlation with eye dominance. So essentially, the more dominant the dominant eye is, the lower the GABA released from the non-dominant eye. So essentially saying, if the non-dominant eye is not very good at inhibiting the dominant eye, then you get this imbalance in between your two eyes. And the nice thing about this experiment as well is that we can also look at the Bolz signal. And if we look at the Bolz signal when you stimulate the dominant eye versus the non-dominant eye, basically what you can see is there is very little difference in the amount of Bolz signal in that case. And not surprising, therefore, if you look at the correlation between eye dominance and the Bolz signal to the dominant and non-dominant eye, they are not correlated either. So it doesn't look like the Bolz signal reflects the eye dominance in this case. So to summarize that part then, so GABA itself doesn't appear to, like the amount of GABA doesn't appear to reflect the overall strength of the neural response, but the GABA does correlate with eye dominance. And this is the case following both this short-term eye patching and during visual stimulation in terms of eye dominance, natural eye dominance. But these studies do have relatively small numbers, so would be worth replicating, but also probably worth building on to add to the amblyopia, to look to see whether it extends to amblyopia, because there is a study by Dennis, who I think is there, showing that there is some correlation as well of GABA in amblyopia, but it would be worth doing that with larger populations. So yes, the next part, which is very quick, is looking at whether the neurochemistry, now not in V1, but what happens to the neurochemistry in area MT when V1 has been damaged by a stroke. So you're all very familiar with this. So this is primary visual cortex. If we have a stroke that affects the primary visual cortex, essentially what you're going to do is going to lose the vision on one side of the world. And this is work done by Hannah Willis that's been recently published. And what you can see here, this is sort of real data showing the stroke in the left hemisphere here, which leads to visual fields that essentially look like this. So they've got a pure sort of hemianopia. Now we know, though, that even when people have got a hemianopia, they are capable of some sort of residual vision quite often. And what we did was we measured the level of residual vision across a group of people with hemianopia. There's about 20 people or 19 people in this study. And what we did was essentially we got them to fixate on this cross. We played a tone, and we had this first interval where a Gabor patch might appear. Then we played a second tone, and we had a second interval. And what they had to say is which interval did the stimulus appear. So it's a two alternative force choice task. And what you see is sort of in this blue line, if we make the stimulus contrast 50 or 100%, this group of people are actually quite good at doing it, whereas people down here who would say don't have any residual vision don't get above chance performance. So we can use this to quantify behavior. So one of the questions would be, why MT? Well, we're looking at MT because actually, if you present a visual stimulus to someone lying in a scanner who's got hemianopia, you present it in the blind field, actually what you see is that you get activity in MT, particularly if it's a moving stimulus. Obviously, it's nowhere near the amount of activity that you get if you present it in the sighted field, but we know that there is activity in MT. So one of the questions is, so is there something that differs in people who have residual vision compared to those who don't have residual vision about the functioning of MT? So what we did was we looked, so what we did was put a voxel to measure our spectroscopy in MT. And you can see this is the average position across the 19 participants. So it's pretty well located in MT, and the black line is a definition taken from an atlas. And we're pretty happy that that's in the right location. And then we had a control voxel, which is sort of on the border of S1, M1. And so it's slightly low down because actually you can't have your voxel too close to the edge of the brain. And we measured the spectroscopy, we measured the spectra in both of those. So this was done at 3T, so we used an edited sequence, which allows us to better visualize both glutamate and GABA.
You're finishing up, right?
Yes, I am. Okay, so what we find is actually in MT, we get an inverse correlation with behavioral performance. So essentially, the better your behavioral performance, the lower the levels of GABA in MT. And we also find that in glutamate. However, not surprisingly, there's no relationship in M1. And potentially unexpectedly, again, there's no relationship between the bold signal in MT and behavior, suggesting again that maybe the neurochemistry is a better marker of performance. So to conclude then, so changes in neurochemistry are quite subtle and require very high quality data. It's important to understand the relationship between GABA and glutamate because you've probably seen that they're quite highly correlated, which we wouldn't necessarily expect. I've shown you sort of further evidence that maybe the neurochemistry better reflects behavior than the bold signal. And what the results could do is actually provide potential targets for pharmacological intervention that might increase plasticity, both for sort of stroke rehabilitation, but also for conditions like amblyopia. And these are all the people who did the work. Thank you for listening.
Very much. Do we have a couple of quick questions for Holly? Yeah. Lovely, Maribeth.
Hi, Holly. This is Lotfi Maribeth from Boston. One of your studies that I've always found really, really important and interesting was your work with congenital anophthalmia. And I understand there are a lot of technical challenges with MRS and making group comparisons. But if you could recruit those subjects again, what do you think you would see in early visual cortex or other visual cortical areas in terms of MRS?
Good question. So we did do MRS in those people, but actually our techniques for measuring GABA and glutamate weren't as good then. I think I only also did work with the congenitally blind people. So we concentrated on sort of choline, which was increased, suggesting that the primary visual cortex was sort of immature cortex. So it was more like sort of non-primary visual cortex. Now we would be able to get better signals. What would I predict? I think so. I guess I would maybe predict that the GABA glutamate levels would be more like for me, I guess, auditory cortex. Yeah, it's a difficult question because it is definitely immature and we can see that. But what that immaturity, I guess we might expect that it would have lower GABA, maybe meaning that it could still be more plastic. But yeah, I don't know. We still need to measure that.
Hi, Holly. This is Frank Tong. Enjoyed your talk, especially since I'm an early proponent of linking binocular ivory with intraocular competition in V1. I have sort of a technical question and a conceptual question for you. The technical being, how much do we know about GABA MRS and its sensitivity to GABA when it's interstitial versus in the neuron? And the more conceptual question is, why do you think GABA levels are changing during deprivation or eye patching? What do you think the process, neuronal process or absence of process is that underlies that change in GABA level?
Yeah, very good questions. So in terms of what we can measure, so my understanding is essentially it becomes visible sort of predominantly on release, but I think that still requires some further sort of investigation. In terms of the temporary deprivation, why it should drop at that point, I guess we're imagining that that basically when you deprive, I don't know if you've done any of the deprivation experiments, but when you take the patch off, it's almost like you feel blind in your deprived eye. So I think we think that would just be releasing less glutamate because it's less active. And so because your dominant eye is the one that's being patched, it releases less glutamate, so you get this reduction because only one of the eyes is sort of stimulating that release of GABA at that point.